Thank you so much for the introduction, and I want to thank the organizers so much for having me and providing me with this fantastic opportunity to talk about the work that I've been doing in my postdoc for the last 2 years. And I'm extremely excited to talk to you guys about viral evolution and the work that we're doing. So to begin, I'm working with Moloney murine leukemia viruses. And the murine leukemia viruses are evolutionarily old retroviruses. More specifically, though, because our lab works with HIV-1, we're looking at this because it is a distant relative of HIV. It causes leukemia, lymphoma, and neurological diseases in rodents, and has been studied since the 50s as a model for retroviruses because around this time it wasn't particularly safe for people to work with HIV. So we use this as a model to help understand retroviral genome packaging and retroviral evolution. But before I dive into MLV, I must first talk about HIV-1 and why I'm studying MLV. So our lab works specifically with HIV-1, and something that our lab has been looking at is the RNA of HIV-1. More specifically, HIV-1 contains a heterogeneous transcriptional start site, meaning that it contains two different pools of RNA. One pool begins with 3 guanosines and the 5' cap, and another pool begins with 1 guanosine and the 5' cap. And what's particularly interesting about these two pools is that one pool functions as messenger RNA, specifically the 3G cap, and it goes on to be translated into more HIV proteins, while the other pool, the cap1G, goes on to be packaged into virus particles and functions as genomic RNA. Further investigating this in our lab in the past, something that Dr. Josh Brown from our lab discovered is that the cap3G more specifically functions as a monomer. It exists as a monomer in a cell, where this red here, the 5' cap, is exposed, and therefore it is accessible for the translation machinery, thus driving that translation. However, the cap1G is primarily dimeric in the cell and in vitro, and this cap here again in red is sequestered between the TAR and poly A hairpins. Because that cap is sequestered, it is not accessible for the translation machinery, and it is thus packaged into more virions, functioning as genomic RNA. Dr. Peng-Fei also went on to discover that not only does this keep the cap not accessible to the translation machinery, he also showed that if this cap is not sequestered, you will not get packaging of that genome. So the cap was really, really important in HIV-1, and this heterogeneous transcriptional start site is really important. But why are we studying MLV? Recent research by Dr. Alice Telesnitsky at the University of Michigan, who is a collaborator of ours, shows that unlike HIV-1, Moloney murine leukemia virus only contains one pool of RNA or a unique start site. And this is going to be a capped 1G RNA. Although we only have one pool, this pool still functions as both genomic RNA and messenger RNA. So the goal of this work is to investigate how viruses can serve functional mechanisms across evolution beyond just genome sequence and protein sequence, because we know that structure and sequence are typically studied here as far as conservation, but we want to look at the conservation of function between retroviruses. So because we want to look at conservation of function as a study of structure, we first wanted to study the monomer-dimer equilibrium of the MLV leader. Again, the HIV-1 has two different pools, where one pool is monomeric and one pool is dimeric. But because we only have one pool, we wanted to understand exactly how this RNA dimerizes. We first did this study where we had our RNA sequence in water. This RNA should be linear, therefore traveling as the monomer would. And we then introduced salt at physiological conditions, and we increased our incubation time. As we increase our incubation time, we notice the appearance of a slower running band indicating an increase in size, which we conclude to be the dimer. And so for this RNA, after about 24 hours, we reach equilibrium at dimer. So we conclude from this that like the HIV-1 cap1G RNA, the MLV leader reaches equilibrium at dimer in vitro. Next, we wanted to explore the state of the cap in the two different conformations. Because the HIV-1 cap1G exists as a dimer and is packaged, and the HIV-1 cap2G and 3G exists as a monomer and is translated, we want to investigate these two different conformations in our MLV RNA. So to study this, we use the EIF4E, or the eukaryotic initiation factor 4E, which we know binds the 5' cap in messenger RNAs. Using this system, We can then introduce our RNA that is not capped as a control, or just RNA that is capped with no protein. With the introduction of the EIF4E protein, if the cap is exposed, we would expect a shift upward or a slower migration on the gel. And in the case of a sequestered cap, dimer, or just a sequestered cap in general, we would expect to not see a super shift indicating that we have binding to two species. Because when I show my dimerization gels, we see that we get dimerization of our RNA fairly quickly. We wanted to first study the monomer, which we had to lock into the monomer conformation first to make sure that we had monomer. We did this by taking the dimer promoting regions of our RNA and mutating in GAGA and GUGA tetraloops to lock it in a monomeric conformation, as we can see here. Upon incubation at 24 hours, we don't get much dimer here, indicating that we had locked it into the monomeric conformation. We then introduced the EIF4E cap binding protein with this locked monomer. As we titrated in our EIF4E protein, we noticed the appearance of a second band that's migrating slower, which we believe to be bound to the RNA, the monomeric RNA. But again, this was a locked monomer, and we also would like to study the native monomer of that MLV RNA. So we repeated this study with the capped native monomer that no longer have these dimer-promoted regions, so we should get dimerization at these hairpins. And in those conditions, as we introduced EIF4E and titrated it in, we again noticed an appearance of that slower migrating band, indicating that the EIF4E protein is binding to the monomeric RNA. We also did this in the presence of an uncapped control, just to be sure that we have specificity for the cap, and we don't see any migration here. So our preliminary data suggests that, like the HIV1 cap3G and cap2G RNAs, the MLV leader contains a cap-exposed monomer. We then wanted to study this with the dimeric RNA. But when we're looking at our monomer, we don't get a lot of separation for the bound and unbound. And therefore, we feared that with the wild-type EIF4E and the dimer, we wouldn't see a shift at all. So in other words, we feared that if we had the dimer bound to the EIF4E, we wouldn't see a shift. Therefore, we wouldn't know if the cap was sequestered or if there was no binding. So we engineered our EIF4E with an EGFP tag attached onto it. And therefore, instead of looking for a shift, we would look for the green signal. So again, because we want to study the dimer, going back to our dimerization gel, we chose conditions. We pre-incubated our RNA and chose conditions that would give us primarily monomer, mixes of monomer and dimer, and then primarily dimer. And we performed this EMSA again with our EGFP EIF4E. The first gel is going to be the athenium bromide channel. Athenium bromide stains only nucleic acids, therefore we should only see RNA on the gel. We then imaged again in the blue channel for EGFP, where we should see only our protein bands now, our protein signals. And then we merged these together. So again, we first pre-incubated our RNA at 0 hours. This should give us primarily monomer. 30 minutes, 2 hours, 5 hours should give us a mix of both. And then 24 hours where we should get dimer. And we then introduce our EIF4E. If you look into the merge channel, we notice that when we have primarily monomer, as we expected, we have binding to our monomer. As we start getting dimer, we notice that we have binding to 2 different species. However, as we reach equilibrium at dimer, we notice little to no interaction with that dimer. Our preliminary data here suggests that, like the HIV1 cap1G, the MLV leader may contain a sequestered cap, the dimer. So in conclusion, thus far, our preliminary data suggests that, like the HIV1 cap1G RNA, the MLV leader reaches equilibrium at dimer in vitro. Our preliminary data also suggests that, like the HIV1 cap3G and cap2G RNAs, the MLV leader contains a cap-exposed monomer. And our preliminary data also suggests that, like the HIV1 cap1G RNA, the MLV leader contains a cap-sequestered dimer. So one pool of RNA is still seeming to have the function of both pools for HIV1's RNA. For the future work, our immediate future steps are to improve the capping efficiency. And because we have so much unbound RNA there, we are unsure if this unbound RNA is uncapped or if the monomer has a secondary structure that has a sequestered cap. Therefore, we are working on methods to improve our capping efficiency to ensure that we have full capping. Next, we would like to explore the role of a cap on viral genome packaging and compare that to HIV1. And lastly, we would like to confirm the state of the cap via nuclear magnetic resonance because our gels can just give an indication, but we want to confirm this with NMR. With that, I would like to thank everyone for listening. I would also like to acknowledge my advisor, Dr. Michael Summers, other lab members as well as our administration. I would love to give a special thanks to my undergraduate trainees. All of these gels that you see, they've done these, and they've done fantastic work. And I would love to thank our funding agencies, the NIH, HHMI, and UMBC. And I thank you all for listening. If you'd like to contact me, my email is here. Thank you.